Hello everyone, welcome to the last webinar of the Gelomics webinar series in 2023. My name is Christoph Meinert. I'm a co-founder and the CEO of Gelomics. For those of you who just don't know us just yet, we're a biotech startup based in Brisbane, Australia, and our mission is really to improve the translational value of cell and organoid-based models in drug development and biomedical research development. Really, our mission and vision is to reduce the reliance on animal models in the field and really fast track the development of life-saving treatments for the patients who really need it. It's my absolute pleasure to host today's webinar and welcome our two fantastic guest speakers, Professor Ferry Melchels and Dr. Cathal O'Connell. Thank you, Ferry and Cathal, for joining today. Very excited to have you here. Thanks also to all the webinar attendees from all around the world. I can see we've got guests from Japan, from Europe, Australia, and across the Asian Pacific region as well. So thanks so much for joining. Very excited to have you here today. So just before we get started with our first presentation, just a few quick housekeeping items. So should you experience any issues whatsoever with this broadcast, with the presentations, please let us know using the chat function and our team will try to rectify this ASAP. We also encourage you, of course, to answer or ask questions throughout the presentation. So if you have anything that's really burning you'd like to ask our speakers, please feel free to use the question sections in the GoToWebinar interface and start typing your questions already during the presentations. And following the presentation, we will have a Q&A session with each of our speakers as well, where they get to answer your questions as well. So without further ado, I'd like to introduce you to our first speaker, who is Professor Ferry Melchels. So Ferry is a professor in biomaterials and tissue engineering at the University of South Australia. He's focused on advancing the field of polymeric biomaterials and pioneers the integration of novel hydrogels, scaffold materials, and cells with innovative biofabrication technologies to create 3D tissue constructs for use both in regenerative medicine and in vitro models. Ferry also explores the development of reliable drug and vaccine delivery devices, opening new avenues in treatment methodologies. Committed to pushing boundaries, his future ambition is to refine biomaterials-based technology platforms, particularly within the tissue engineering and drug delivery community. Recognized for his significant contributions and expertise, Ferry's work has the potential for groundbreaking advancements in medical treatments and patient outcomes. So Ferry has published over 60 research papers and they have been cited over 15,000 times. So clearly a lot of impact in the field, and we're really happy to have you here today. I personally have known Ferry for a long time, since approximately end of 2011, when he was actually a postdoc at the Queensland University of Technology. At the time, I did my undergraduate studies here in Brisbane and bumped into Ferry at the time. So Ferry already in 2011 was one of the very early users and pioneers in the development and application of gelatin methacrylol hydrogels, or better known as GELMA hydrogels, which are now biomaterials and are now widely used across the tissue engineering community globally as well. And so I'm really looking forward to your presentation today, Ferry. If you could just make sure you share your screen with our audience and we can get started as well. So without further ado, I'll hand over to Ferry for his presentation.
Oh, yes, thank you, Christoph. There we go, show my screen. Sorry, done for me. Are you seeing the right screen? Type of screen?
Yes, we can see the right screen. I think we're all good to go. Thanks, Ferry.
Excellent. Okay, thanks very much for that wonderful introduction, Christoph. Yes, I recently joined the University of South Australia only 3 months ago, so I'm fresh off the boat, as they say. I'm advertising currently a PhD studentship, looking for the first PhD candidate to join the lab here that I'm starting to build. Unfortunately, it's only the scholarship covers only domestic students, but if there are any Australian citizens or permanent residents or New Zealand citizens that would like to take on a PhD in biomaterials, please contact me for more information. So let's start gelatin. It's actually a bit more than a decade. It's about 13 years and 7 months, but it doesn't sound as nice as in the title, so we made it a decade. I'm going to talk a bit about the early days of GELMA research, some physical properties, characterization, its use in biofabrication, and then a few different tissues that we've engineered with it. So did a bit of a deep dive into the history of GELMA. So it's a bit of a rabbit hole every time you you go on Scopus and look back at what's been done, but it was quite interesting to see. So the first report of GELMA being synthesized and characterized was back in 2000 by this group from Belgium, polymer chemistry group in Ghent. They reported on synthesis of gelatin functionalized with methacrylic anhydride. And so that kind of started the field. What many people don't realize, actually, they use gelatin type B, whereas almost everyone else has been using type A. And sometimes that also leads to some confusion of properties, other things, other observations people made. And also interesting is that they suggested it would be used for drug delivery or wound dressings. They at the time didn't realize yet the opportunity for 3D cell culture, which we're using it now a lot as for. So actually, after that paper, not much happened for quite a bit of time. I did my PhD around this time, the second half of that decade. And I was started to think a bit about using GELMA, but I never really got to it until I was at QET at IBI in 2010 as a postdoc. And then there was a fire alarm. So we all had to go outside, standing outside, needed to evacuate the building. We just, getting a little bit bored. So I just walked over to Woolworths just across the road, the supermarket, and got a bit of gelatin there just out of the supermarket. And that was actually the start of GELMA research in Brisbane. So I took that back to the lab, and the first next Friday afternoon, that's the time when you try new things, I made my first batch of GELMA. Only a few months after that, this paper came out from the Kadam group from MIT. And this is probably what really set off the GELMA research. You can see I put in the number of citations more as a matter of a measure of impact of a certain work. So they did some extensive characterization and showed cell encapsulation as well, which really kicked off the GELMA research. But in the meantime, a few other things had been done as well in between since that 2000 paper. So these people in Italy used GELMA to make polyhype scaffolds. So these high internal phase emulsion foams as scaffolds, and seeded cells in the scaffold afterwards. So it's not really as a gel, but more as a scaffold, which didn't get too much attention. Then these two papers, 2007, didn't generate too much attention either, but they had used GELMA as well. And then in 2009, there were actually two papers who started using cells within the GELMA, encapsulated cells within GELMA hydrogels. And so that's the work that kind of predates that big paper, but put it really on the map. And then 2011, there was another paper from the Belgian group from Ghent, together with the TU and Vienna, who used it into photopolymerization. So that's a bit of the early days of GELMA. I'd like to just talk a bit about the name GELMA and so the abbreviation GELMA and the other names that it has and has had. So the first paper was talking about methacrylamide modified gelatin, which I think is quite a good name, because it is mainly methacrylamide groups that you get when you react gelatin with methacrylic anhydride. But interestingly, almost all the papers that followed talked about gelatin methacrylate or methacrylated gelatin, and the 2009 paper was the first to use the abbreviation GELMA. So that came only nine years after the introduction of the material. But there is actually a difference between methacrylamide on the left-hand side and methacrylate on the right-hand side. They also have different reactivity. So one has the nitrogen there. So they're not exactly the same. So actually the methacrylated gelatin didn't seem to be the best name, because also those papers acknowledged it was the NH2 groups that got functionalized. So other groups used a bit more generic names like gel mod, modified gelatin, or cross-linkable gelatin. So in 2014, there was a symposium in Nijmegen, which my hometown back in the Netherlands, and Ali Fademasaini was there as well, and we had this nice dinner in this fortress here at the dikes of the river. And after a few wines, I gathered up the courage to confront him on the name, and I said, "Mate, you're calling it gelatin methacrylate, but it's actually methacrylamide. Right? Methacrylate's not the right name." So what he did as a result, he had his people in the lab do a thorough characterization of the groups on GelMA. And yes, the majority are methacrylamide, but apparently there's also some, maybe 15 to 20% methacrylate groups as well. So actually we have both. We have several different types of groups. And that's why we agreed together, let's call it gelatin methacryloyl, which is a bit of a tongue twister. It's not easy to pronounce, but it covers all of these different types of functionalities you can find on GelMA. So we agreed on that. They coined it in a review 2015. And in 2016, we worked together. You can see Christoph is carved on this paper as well, when we wrote this Nature Protocols paper on GelMA. So you can still use the abbreviation GelMA, which is nice, easy to pronunciate, for gelatin methacryloyl, and published on how to make it, how to characterize it, and how to use it with cells. So much about the history of GelMA. Let's look into physical properties. So we started looking into GelMA and mechanical properties. So when you use a gel for 3D cell culture, the mechanical properties, and particularly the stiffness or compressive modulus, which is stiffness under compression, is quite an important parameter. It has quite a big influence on cell behavior. So we looked at the influence of the concentration of GelMA as well as exposure time. So stiffness would increase with concentration and with prolonged exposure to UV, which obviously that is dependent on the system you use, like your photoinitiator concentration and your light source as well. But we had standardized these conditions in Brisbane, also in Utrecht and Würzburg, and in Christchurch, and the different groups that collaborated all worked on GelMA, so we could compare the results. And we found that these we could really pinpoint the stiffness really well. So here you see the mathematical formula, and the dashed lines represent this, of how the stiffness develops with exposure time and at each concentration. So it's important, but also for natural materials, it's quite tricky, obviously, usually to tailor this really. So we were quite happy that with GelMA, with these, we could really control and reproduce the stiffness as well. So later, when we started using this for cancer models, we synthesized new batches and did the same characterization. You can see here that it's really quite reproducible, which is not that common for natural materials, and so it's quite happy. We were quite happy that we could reproduce this. One other thing that we noticed at the time was that if the gel, the precursor solution, was left to stand for a while before we turn on the light, that actually led to an increase in the modulus. And this is only a small increase here. We didn't give it too much time, but it was a single observation and reported it, but didn't look into it much further at the time. Which later I've been looking into this more, and just so having a GelMA solution, you can either keep it warm and cross-link it straight away. So here, then you get a fully chemically cross-linked gel, or you can first let it gel physically. For example, in the fridge, it goes a lot faster than on the bench. So in the fridge, you will first get a physical gel, as gelatin does, through triple helix formation, and then you can do the UV or visible light, like the photo curing afterwards. So the first GelMA paper kind of said it loses its ability to form a gel if you make it into GelMA, which is almost, which is mostly true for gelatin type B, but Gelma made from gelatin type A still does gel a lot more. And it leads to an enormous increase in stiffness. We see up to 40 times increased stiffness at the same concentration. But if you look at it differently, you can make a gel and create the same stiffness with much less gel mat. So this is quite, it's another parameter, and it's also good to keep in mind when you're making a gel, if you allow it to gel physically, you will get a much higher stiffness. So even strength is increased as well, about 10 times. And we did some DSC to confirm that actually we have this triple helix formation going on. So we see a similar enthalpy, so similar energy released by letting Gelma gel compared to gelatin. Recently, together with Warsaw University of Technology, we compared them, looked at the effect of cells. So it chose a 5% pre-gelled UV visible light photocured and just a straight, so it's the SC. And we saw, despite having exactly the same stiffness, same gelma, that cells do respond very differently. So we saw a lot more stretching of the cells within the double cross-linked gel compared to the single cross-linked gel, or the same stiffness. So that is very interesting. We also looked at diffusion a bit longer ago through this FRAP process with a fluorescent FTSE dextran. Dextran 70 kilodaltons is quite a big molecule. Despite that, we saw that the diffusion coefficients were quite high. So, for example, looking at the 5%, for example, still diffuses at more than half the rate compared to still water. So really these gels, although they seem to be solid, because it's so much water, actually even big molecules diffuse really fast through it. So to those molecules, it's almost the same as being in water. So they hardly notice the polymer chains being there, which kind of means that you can use this for 3D cell culture. So now we've looked a bit into those physical properties. Let's look at biofabrication using Gelma for fabricating constructs. We still have this dilemma when we're fabricating or printing with gels, that cells typically tend to favor soft gels, snotty, low polymer concentrations, cross-link densities. But if you want to build a construct with a high shape fidelity, you want something stiff and a lot of polymer and not too much water. So there's always a trade-off, or you find strategies to get the best of both sides. So 3 different strategies we follow are to optimize rheology of the gel itself, or to use internal supports, or to print into suspension media. So with optimizing rheology, you can add different components. So our first try was hyaluronic acid, which increases the viscosity a lot. So that helped to get it from dripping from a needle to actually making a filament. It's more viscous, it still keeps flowing. So you still need to be pretty quick with photocuring to not lose the fidelity of the structure you've printed. So after the later looked into gelongum, it's a polysaccharide that can cross-link ionically, can make a weak ionic bond that can be overcome by pushing it through a needle. So by tweaking the gelma and gelong concentrations with salt, I was able to get the right rheology to print these. The top ones are molded, but you can see these are not photocured yet. Also, the bottom constructs, they aren't a solid pyramid and that porous structure. So the rheology here is optimized so you can build a structure, and even before photocuring, it already keeps its shape. So that is because of that ionic cross-link between the gelongum bonds that's broken up when you push it through the nozzle. You also get shear thinning, so it becomes more liquid to get it through the nozzle. But as soon as it leaves the nozzle, that ionic network is reestablished and it becomes a gel instantly. And then you can do a second step of photo curing, so you can take it to prolonged culture. Later, in collaboration with the Würzburg people, we looked at the more generic, generalized these rules of rheology, of what is the rheology required for good bioprinting, and I kind of wrote this white paper that has been quite a good reference for people starting with bioprinting gels to look at what are the rheological parameters required. Internal supports is a second strategy where you kind of just use two materials. If you have a material that supports the structure, then you can print the gel with the cells. It doesn't really matter that much what is rheology, so what will be kept in place by that reinforcing material. So we used Gelomics. Poloxamide is quite a widely used and known material because it has a really good rheology, easily printable. We modified it with methacrylate end group so we could cross-link it, but also with a small oligoester link, like a degradable link, to make it hydrolyzable. And then we could print this together with the bioink with the cells, and the bioink was just plain Gelma 37 degrees liquid to fill the gaps. So here we use Fitzy Gelma, so you can see the Gelma in green with the reinforcing ink. And by using more reinforcing ink, you can get a higher stiffness. And that you can see here that it really keeps the shape. So this is it's about a 50-50 construct, and you can print shapes and build up to higher structures despite the gel being not self-supportive. And that degradable link, by using different types of it, so caprolactone is slowly degrading, lactide faster, and glycolide even faster, we could make this gel to degrade at different rates. So you can tweak how fast you want to degrade this gel to degrade, so to match tissue formation. Another strategy that we use is fiber reinforcement with melt electrowriting. So by melt electrowriting, we use electrospinning without a solvent, so we get quite a stable jet and collect this on a computer-controlled collector. So you get these in the tens of micron-sized type fibers. These scaffolds with really high porosity, so you need only a few % of polymer to create a scaffold, which is a bit different from the typical FDM fused deposition modeling scaffolds, which have much thicker struts and also lower porosities. With this, so we made these scaffolds and infused them with Gelma, cured the Gelma, and then with this we could get quite close to the mechanical properties of native cartilage. So you can see at the bottom two curves are just plain Gelma, or the plain scaffold, but the combination of them works synergistically and makes this this reinforced Gelma that is both stiff, a lot stiffer, so approaches the stiffness of cartilage, but it's also compliant, so you can compress it much more than you can a typically an FDM printed PCL scaffold. So the 84% there is an FDM scaffold at the highest porosity we could attain, which is actually, it's too stiff, but also at 10% so it fails. So that's not the best scaffold for cartilage. So these fibers really helps to reinforce the gel and make something with similar mechanical behavior to cartilage. Same thing set here, basically. Then third strategy, suspension media. That's something which has really boomed over the last few years. So this is quite a nice video from a group in the US who pioneered this. So here you don't print on a surface in air, but you print into a bath, kind of a transparent bath here, which is you print into a gel. So that gel is typically, or in most cases, is a granular gel. So it's like a ball pool, small micro gels that are packed, jammed in the space. So that's why they're slightly solid, and then they suspend things. If you put something in there, it will stay in place, it won't sink. but it can still move, so the nozzle can travel through, push these aside, and you can deposit things. So you get this liquid solid transition. And we've been making these out of gel and gum. So again, gel and gum, we've used it before as a rheology modifier. Here we've used it to make a granular gel. And you see that it does have a yield stress. So it goes from a solid when it's not sheared or a very low shear stress. And then if you overcome the shear stress, then it turns liquid. But it's reversible, as you can see in the bottom figure. So you're going from high to low stress. It reestablishes itself. So here, when printing into it, we could attain much higher printing speeds than on a surface, up to 60 millimeters per second. And interestingly as well, if you slow down on a surface, the gel will just spread and make a wider filament. But here it actually starts meandering a bit in the suspension media. So you can create this tortuosity, which might be interesting for some specific models if you wanted to model a gut or something else, that this tortuosity might be quite interesting. So we could also print with droplet printing, with the cells in droplets, and kept them alive for over a week, and also printing different cell populations close to each other to make multicellular constructs in the suspension media. So you might think, but that suspension media, that's not Gelma, that's gel and gum. True, that is how we use it first at the top here, but then we also made a photocurable one combining the gel and gum particles, the granular gel, with Gelma. So this suspension media can be cured afterwards. So our idea here for future use is that you cure you don't have to print the bulk tissue. So the Gelma suspension bath can be your bulk tissue. You can print cells or vasculature by printing sacrificial material and removing it afterwards. Then you can cure the bulk, and you've got all these printed features in them. So you don't have to print the big bulk of the tissue, and it can save a lot of time, increase fabrication speed and throughput. Gonna take a sip of water now. So just a quick overview of some of the different tissues we have looked at engineering with Gelma. So cartilage is one of the early ones. This work is with Peter Levitt, who's a co-founder of Gelomics, and we put primary chondrocytes into Gelma and into a few other photocross-linkable gels and methylated gels. And on the top left, you can see within the Gelma the cells spread really well. They proliferated well, and you see them spreading. The spreading is not a good thing for cartilage because it means it's dedifferentiated. We see they secrete collagen type 1, which is not the right type for cartilage. They did better in hyaluronic acid, where they stayed rounded and they made the right types of collagen and gags. So that's why we liked the fact that proliferated in the Gelma. So we combined it afterwards in the second study, Gelma with HA, and found a good combination where we saw the cells proliferating as well as creating tissue of the right kind, the right gags and collagens. And here on the right, you can see particularly the GHA, that's a Gelma with HAMA. We can really see the compressive modulus increasing with culture time as a result of the neo cartilage that was formed. So obviously you have to, whenever you change the Gelma, the ink for biofabrication, you have to also look at the effect on cells. So we used also the fiber reinforced gels that I talked about before. We used them with chondrocytes, and so we could still engineer that tissue, have the cells proliferate and make tissue in those gels. They didn't really weren't really affected much by the fibers. Whereas when you put gel on gum through the gel to modify the rheology, make it more printable, you see a completely different picture. So you always have to keep in mind when you improve the printability, you have to look at the effects on the biology as well, which probably then advocates for using one of those other strategies of using an internal support of suspension media, so you don't have to tweak the bioink that much. In terms of vasculature, blood vessels, we've looked at the capillary formation within Gelma, and saw we really needed really low stiffness, below 2 kilopascals. We saw the capillaries forming. As soon as you get to 3 kilopascals, you get 80% DOF. The capillaries no longer formed inside the gel, so really soft gels. But then when we culture UVEX, so endothelial cells on top of Gelma, it shouldn't be too soft because then they don't adhere, but they let loose. So you need a minimum stiffness. So we found this soft spot here, the 50% DOF, 2 kilopascal stiffness, that supported both capillary formation inside as well as monolayer of endothelial cells on the surface. And so that we took that one forward to make this very simple model, just a block of gel. In the bulk, we saw capillaries forming. We see that endothelial cells had lines, a channel in the middle, so you can see that lining. And we even saw anastomosis, so the capillaries would connect to that endothelial lining in that channel. So that was pretty nice. This is really recent, just submitted last week. Work with the University of Edinburgh, where we used Gelma in combination with solution electrospinning to make blood vessels grafts. So the first printing the Gelma on a mandrel, and then electrospinning a fibrous mesh around it for reinforcement. You can see the Gelma is autofluorescent, so you can see the thicker gel layer and the thinner polymer layer around it. And so we managed to get burst pressures around the same as native arteries, which is the green dashed line. The gel itself would burst at much lower pressures, but the fibers really help to get a good burst pressure. And interestingly as well, the compliance is similar to native vessels as well. On the right, those are three formulations we work with. On the left is a lot of natural arteries, and the Dacron and PTFE, those are things used in the clinic for blood vessel grafts, but these are much stiffer, so their compliance is a lot lower. So we managed to get much closer to native blood vessel properties, and could again seed Gelma endothelial cells on Gelma to endothelialize. Cancer models, this is back from the QUT days, the early days of using Gelma. So we used different concentrations, which means different stiffnesses, as we've seen before, and saw different behaviors. So if it's very soft, 2.5%, we see a bit of spreading in individual cells or a bit of chunks of cells. But at the higher stiffnesses, we see nice spheroid formation, which is a good model to test then cancer drugs on. So we saw proliferation, and I think I'm just going to go a little bit quicker here in the interest of time. And the Gelma was also shown to be a good vehicle to establish an animal model, so we could more reliably establish a tumor in mice than with just injecting just cancer cells, as was done often. And more recently at Harriet Watts, we did some printing of colonies of cancer cells with the droplet printing using Gelma in a matrigel matrix, and see how they grow towards each other. So with the idea of looking at next steps of different cell types and see how they interact. So my half hour is up. I'd like to say thank you to a couple of, these are the main people have worked together with on Gelma over the years. Might have missed out a few. Apologies for that. Some of the funders, I actually realized when I made this that some of the early work was all funded by the EU, and a lot of the later work is actually not through my own grants, but working with lots of people who saw the value of working with Gelma and approached me to work with it. So thank you to Gelomics for the invitation, and thank you all for your attention.
Thanks, Ferry. Fantastic talk. Really good to hear the story of Gelma, and you've been around for a long time in the field and definitely made some fantastic progress there. Thank you so much. So just a quick reminder to everyone, if you have any questions, now is the time to put them through our questions section on the webinar interface. We've got a few that we can already ask, Ferry, if you're ready to take those. So first is actually from Cathal, who will be talking in a few minutes as well. So he says, very awesome talk, long time listener, first time caller. What's your favorite photo initiator and why?
My favorite photo initiator? I would probably say LAP is my favorite. We use a lot of the EGA-QR 2959. And I think generally in research, people stick too long with what they're comfortable with and used to. And that photo initiator was only used because that one paper back in 2000 in the ENSA Christianity group, Stephanie Bryan just tested a few off-the-shelf initiators. None of them were intended to ever be used with cells. One of them turned out to be quite reasonably well water-soluble and less toxic than all the rest. And after that, just people kept using the EGA-QR, but we also don't really know the long-term effects of the UV. Even if the viability is good after 24 hours, we don't know what it does to DNA damage. So I'm really a fan of using visible light. I like the ruthenium system as well that Kun Lim in Sydney uses, but that's particularly if you make it, you have to use it pretty quickly because it does, it doesn't last too long. But LAP has now become more widely available commercially as well, so it's easy. You can use these 4 or 5 nanometer light sources. So a lot of printers use that light wavelength, and it's a lot safer for the cells as well. So I would say LAP. It's my favorite.
Yeah, I'll also comment on that as well. Our experiences as well in the lab. I mean, we've also of course started with UV cross-linking 365 nanometer, and then switched to more visible wavelengths. And we actually also found that particularly sensitive cell types just wouldn't behave as you would expect when they were cross-linked using 365 nanometer light with EGA-QR. And we never were quite sure if it's really the UV or if it's a photo initiator. But either way, from a practical perspective, we found that sensitive cell types, primary cells, also HUVECs and similar cells actually had much better phenotypes when cross-linked with a visible light photo initiator.
Yeah. An additional advantage is is less sensitive to oxygen inhibition as well. So the 365, the EGA-QR, you have to create enough radicals to overcome the oxygen that diffuses in and inhibits it, and to get that balance right and make it reproducible is a lot harder as well. So yeah, definitely.
I think there's a lot of work in the field also to really try and redshift the photo initiator spectrum even more to have be more in the visible light range as well.
Yeah, no,
thanks, Ferry. So a couple more questions, if you don't mind. I'll just read them out one by one. Next question from one of our attendees says, great talk, Ferry. Thank you very much for the detailed history of Gelma. The physical gelling before photo cross-linking is a very interesting one. How stable is the physical cross-linking in the photo cross-link Gelma at high temperature, for example, 37 degrees? Is the mechanical enhancement maintained?
Yes, it is. Yes, so it is. What I think happens is the physical cross-linking kind of creates a template that is locked in by the chemical cross-linking in the second step. So yes, I've gone up to 60 degrees or so, and it doesn't lose it. So it doesn't lose the stability. Actually, it does lose the thermosensitivity. And I skipped over one because in the interest of time. But I actually now we're talking about it. I'm just quickly going back to it.
Yeah.
It is an interesting one. Here, this one. So here you see that the bottom line is just rheology on physical cross-linking of GELMA. So how the shear modulus builds up over time at 5 degrees, and then the chemical crosslink, so the single chemical crosslink. If you add that up, like the 8 kilopascals, so that the second, the top line, the PC plus SC, this kind of a hypothetical value what you get if you have the physical cross-linking plus the chemical cross-linking of just a direct cross-link without. But then you see the measured points actually. That's what you get with double cross-linking, doing one step after the other, and you can see how it is really a synergy. So it's not just adding the two effects together, but they actually work synergistically to get a much higher stiffness. And it's about 1 hour, and the fridge is about the time where this starts plateauing. So that's also to not, that's probably a good time then. That's what we took forward with the cells as well. And most cell types are quite happy for an hour in the fridge.
Okay, and have you seen any differences in cell response when you have these partially formed, I guess, collagen fibrils that you get through thermal gelation of GELMA? I mean, of course, it's difficult just to really differentiate and have a clear understanding of this because it's always tied to also different mechanical properties or stiffer properties, but perhaps you've seen.
So these cells, these are human, the bone marrow-derived stem cells. They are, the top ones are in the double cross-linked gel, and the bottom ones in the directly cross-linked, single cross-linked chemical gel of the same stiffness. So really, our hypothesis was also just people tend to focus on stiffness, and yes, stiffness is very important, but it's definitely not the only thing, because these two gels are the same composition and the same stiffness. So the microarchitecture definitely has a very big effect as well. So we see the cells are much more, they can stretch, and we also saw them migrate. So migrate at the edges, they would migrate out of the gel. So they're much more mobile in these double cross-link gels.
Okay, fantastic. Let's continue with the next question. Again, from an attendee. Thanks for your great talk. To more closely mimic the native tissue microenvironment utilizing hydrogels, how important is the focus on viscoelastic or time-dependent characteristics of the hydrogel in line with other characteristics such as stiffness? So basically, what is the importance of viscoelasticity versus stiffness?
I think it is important. It is, last few years has been shown as well that not looking just at the stiffness but more at the viscoelasticity, and it's been shown that in viscoelastic gels you get different behavior of cells than purely elastic. On the other hand, also most tissues have about the same viscoelasticity, as in there's like a 90% elastic, 10% viscous component. So it's not like we have a very huge range. So I think also that viscoelasticity, that's kind of a phenomenal measurement. You measure the mechanical properties, which are a result of the microstructure, and the cell behavior is also a result of the microstructure. So although, so they're both a result of the microstructure. So I don't think you can say this viscoelastic property or behavior will lead to this kind of cell behavior. But it's definitely an important measurement to do. But then there will be, similar to here, same stiffness, different behavior. There will be gels that have the same viscoelasticity and still evoke a very different cell response. So you have to look at it from different angles and really kind of think like the cell. What does the cell see and feel?
Perfect, thanks, Ferry. We've got 3 more questions that we might go through today. Next question is, what, if any, are the translated applications of GELMA to this day? So do you know of any application in the clinic that's relating to GELMA?
I'm not aware. I think, and it's really pity that that a lot of the research stops at the preclinical stage. So it's often small animals, and then it does stop there. So there are a few things that aren't discussed often, which I think are quite important and might be barriers for GELMA clinical translation, which is the presence of endotoxins that can really evoke immune responses and really hamper tissue regeneration. So with immunodeficient mice, you don't see that, but as soon as you move to immunocompetent animals or people, human, it could be a very different story. And there's a few indications that that might be a bit problematic. On the other hand, there are other applications of gelatin as tissue adhesives and a few surgical applications of gelatin. So there is some potential there. Otherwise, these materials wouldn't be used in the clinic. But and then toxicity, cytotoxicity is completely different to like systems toxicity. That some materials are used in the clinic that wouldn't be so good for cells, but for the organism as a whole, it does fix the problem. So that sometimes justifies use. But yeah, there are definitely some challenges there in the clinical translation, particularly in the regeneration.
Yeah, and then just to add on to this, there are companies such as Rousselot Biomedical from Belgium who are now manufacturing reduced endotoxin-free GELMA, so ultra-pure GELMA varieties, and they are really aimed to really fast-track that clinical translation of the biomaterial. So we're also working quite closely with Rousselot on that as well.
Yeah.
Cool. A few more questions. Let's maybe try and answer them fairly quickly, just in the interest of time. Another one from a good friend of yours, who you know very well, our co-founder Peter, who you've also published with. He says, "Hi Ferry, thanks for the great presentation and welcome back to Australia. Can you talk a bit more about how the tubular constructs with the PCL and GELMA are made? Is it the GELMA on the inside or outside of the tube? It'd be great to hear and describe the process a bit more."
Yeah, so it's hi Pete, good to be back, and thanks for your welcoming message. The GELMA is on the inside, so we have a mandrel, that rotating mandrel, and we have a nozzle that prints the GELMA, and the GELMA, so because it's cell-free, we can, I'm just quickly going there, we can, so we kind of keep the GELMA at a certain temperature for a while, so it's in that in-between state where it's very viscous but not yet a gel. So then we can print this on this mandrel, rotating mandrel from bottom to top, and it doesn't sag. We get a nice even thickness, and then the electrospinning is around, so we get a coat like on the outside. So here on the pictures you see it's a bit hard to see, but you see a bit the white sheet of polymer around it, the reinforcement. So the idea is the inside that this can be lined with endothelial cells, so we can have endothelialization, make small diameter blood vessels that won't give clotting, and that can be sutured to blood vessels. And because they have a very similar mechanical properties, and they also dilate together with the rest of the blood vessel to have that same compliance. That's the idea to be a good functional graft. And
does the burst pressure also compare to native arteries?
Yes. So these are burst pressures. The native arteries, the green dashed line is native arteries. Okay. So we had a few different compositions of the polymer, with PCL and PLCL with lactide as well. So we are quite around the same burst pressure as a native artery.
Well, maybe you've got something here you could translate into the clinic. Looks very promising.
Yeah, now I'm talking about it, I think like this is actually pretty cool.
It is pretty awesome, yeah, for sure. Cool. Let's two more questions. Let's quickly go through them as well, just so we've got all of them answered. What was the effect of increasing the concentration of GELMA on cell viability and proliferation?
It depends a lot per cell type. It's very different from cell type to cell type. So endothelial cells, for example, UVEX inside a gel it has to be really soft. You have to be really 2 kilopascal below, or they won't survive. For cartilage, actually, we need a minimum of a 10% or higher to be stiff enough to prevent them from spreading and dedifferentiating. So for some cells, you need to go high, other cells you need to go low. So there's not one answer to that question. So it really depends on the cell type.
Okay. Last question. Thank you for sharing your fascinating research. I'm intrigued by your work on mimicking cartilage using germa and stem cells. I was wondering if you have conducted any compressive tests on these germa constructs with incorporated stem cells. Additionally, I'm curious about your thoughts on how in vitro biomechanics tests might impact the activity of the stem cells. I can also add on to this a little bit as well, because that was part of my PhD project.
I'll let you answer the question, Christoph, if you don't mind.
Sure.
No,
absolutely.
I'll quickly. So we did because the fibers carry most of the weight. It actually doesn't really matter if their cells are not how soft or stiff the gel is. So that's really governed by the fibers. But then I'll let you go on the stimulation of the cells.
I mean, Ferry, Professor Travis Klein from QT, and Peter Levitt, and myself, we've all been working in the cartilage tissue engineering field. And we haven't really focused so much on using stem cells, but primary chondrocytes, so the cartilage cells directly from a donor or patient. And we've incorporated this in germa, sometimes germa with hyaluronic acid. And what we found that these cells would really develop and grow into neocartilage constructs over time quite significantly. So there was a significant accumulation of cartilage-specific proteins and macromolecules. And in terms of mechanical properties, we really saw that these increased quite significantly over time. So within a month, we typically saw something around 2 to 3 times the mechanical properties compared to the initial start date. That's for pure germa gels. In the fiber-reinforced hydrogels, we could really, towards the end of our research, we could really tailor the mechanical properties to be very, very similar to actual native tissues as well. And during my PhD, we've also looked at the effects of mechanical loading on the accumulation of these cartilage-specific protein markers, and we found that these have also improved and increased. So for example, collagen type 2 accumulation, when you apply mechanical loading that simulates joint movement, has really improved and increased quite significantly as well. Same with, I believe, mechanical properties and gene expression as well. So that's kind of in a nutshell, but you know, if the person is interested to hear more about it, we've got a few papers that we can share and also share our thoughts. So I think in the interest of time, Ferry might finish up with a Q&A session here. Thanks so much for your talk again. Really fantastic presentation. Great to have you back in Australia.
So much time, but yeah, it's a pleasure. Thank you.
Thanks so much. So if you don't mind, just quit sharing your screen, and Cathal, if you could come back online, we can continue with your presentation now as well. Thanks so much, Ferry, again. So Cathal, great to have you here. Thanks so much for joining in today. I'll just make you a presenter, but of course, before we get started with you, I've got a short introduction as well, which I'll run through in a second. So Cathal, it's an absolute pleasure to have you here and pleasure to introduce you to our attendees. So Cathal is an expert in biofabrication with a strong background in physics and nanoscience. He's a senior research fellow at RMIT University in Melbourne, Australia, and as a lead biomaterials scientist on the BioPen, which is now, I believe, called the Excelda project. He's working towards a new era in treating knee defects. He has published in prestigious journals such as Nature Nanotech, and beyond the work in the lab, which Cathal is in right now as well, he's made science accessible to really the masses and the public, authoring over 150 popular science articles. His piece, The Mystery of the Missing Antimatter, for example, was featured in the Best Australian Science Writing in 2017. He is recognized as a finalist in the ABC's Top 5 Under 40 in 2015, and has accolades with the Unthinkable Inspire Australia Award. So a pleasure to have you here today, Cathal. Really looking forward to your presentation today. So without further ado, hand over to you. Thanks so much.
Great, thank you. And can you hear me okay?
Yes, we can hear you and see also your presentation. Thank you.
Perfect, perfect. All right, thank you. So I mean, it's really an honor to be speaking alongside like such a research leader and a JAMA pioneer like Ferry. So I appreciate this opportunity to share our work on biofabrication. So I'm employed by RMIT, but most of my work is based at the BioFab 3D lab at St. Vincent's Hospital in Melbourne. So this is a shared lab co-owned by 5 partners. There's 4 universities and the hospital that all work together in this shared space. And we're a biofabrication lab within the hospital site. So we have, doing 3D bioprinting and so on. And we're working directly with clinicians at the hospital, especially orthopedic surgeons who sit in the floor straight above my head right now. So, and this lab is the precursor to a much larger investment, the Aikenhead Centre of Medical Discovery, which is under construction right now. You can see the construction site there. There's even a live webcam all the time if you're interested in following along. But anyway, that will be our 10-story, 400 researcher translational hub here in Melbourne CBD, and that will open start of 2025. So please come and visit if you're here anytime. So at the moment, I'm working with RMIT. I've also been on an industry placement at a medical device company. So this past year, I've been doing a lot more applied industry R&D than academic research, but it's great to share the research that our team has been conducting over the past few years. So some of the research that we do at BioFab, we target various skin, various tissues like muscle regeneration, like in vitro skin models, osteosarcoma models, 3D neuronal modeling, and a few other things. All of our projects pretty much in the center, they all have clinical collaborators, and one of the criteria of the projects is that they have a translational aim or focus. So what I'll talk about today is mostly along in the category of cartilage repair. We had a great introduction to cartilage repair already from Ferry. And the project I've mostly been involved in is called the BioPen project, now Excel the Pen. This started off as like a hardware design type project led by Professor Gordon Wallace and Peter Chung to develop a handheld device for delivering stem cells in surgery for cartilage repair. But now the project has evolved to cover a lot more categories of research, including biomaterials development and stem cell technologies and so on, all geared towards articular cartilage repair. So we have quite a growing and large team now aiming to translate the technology, which we're funded through the Medical Research Future Fund. Most of our work over the past few years, well, 2 or 3 years, has been on generating the patents and working through the design and regulatory and the quality hurdles, the kind of things that Ferry was alluding to earlier. And so we have a team of biologists, material scientists, as well as hardware engineers, and led by orthopedic surgeons and by Professor Gordon Wallace. Because a lot of our research in this umbrella is kind of under protection, I'm not going to talk a lot about the actual cartilage translational projects. Instead, I'm going to talk about some of the materials and fabrication challenges we've had to tackle along the way, and I hope that will be the most interesting and useful thing for this audience too. So, I wanted to talk about things that would be of interest to Gelomics audience, so I've gone back, going back to where I started off, working with photocross-linkable hydrogels a few years ago, well, about 7 years ago, so not quite as long as Ferry. But we were inspired by Ferry's papers, and that's one of the reasons why we started using JAMA back in about 2015 or so. So, we were just starting to use this material, and as an exercise to try and bootstrap some expertise, I decided to try and study the photocross-linking reaction, just try and learn this, how to get the best results with this new material. And the technology the technique I used to explore the photocross-linking reaction itself was in situ photorheology. So often the field focuses on the final properties of the structure, like the compressive modulus, obviously, or tensile testing. Obviously, these are really important. They're the functional properties that are going to define how cells behave and tissues develop. But there's also a lot that you can learn from monitoring the reaction in situ. So, for example, here's the evolution of a typical photocross-linking reaction, where you turn on the UV light, and you can monitor when that reaction starts to occur, and then when it reaches a plateau as it's reaching completion. And already from the shape of the curve, you can learn a lot about what's going on during the reaction. You can learn about the oxygen inhibition by the lag time, so how long it takes before you start to see an increase in stiffness. You can learn about what's going on in terms of the interaction between the polymerizing molecules and other components in the system, by watching the reaction rate, and I'll talk about that later. And you can also monitor how the reaction proceeds when you turn off the light, and that can tell you about the quenching activity of other agents within the system. So there's a lot you can learn by watching the reaction occur as it evolves. So, oh yeah, by the way, so the data I'm talking about here is generated in an areometer, where you've got a quartz plate on the bottom, and you shine up the UV light underneath, and then you can that's how you can monitor the change in stiffness as a function of the light exposure time or intensity and so on. So, at the very start, let's start with the light. So in our field, we often talk about using, say, a 405 nanometer light source or 365 nanometer light, and of course that's a simplification. In reality, every light source emits a spectrum, like a range of wavelengths, and it might be a tight range, or it might be a wide range, depending on the type of light. So this blue part under the curve here is actually the emission spectrum of our LED light source, which is centered in this particular paper. It's centered on 365, and then this gray band and the red one, they show the absorption spectrum of the photoinitiator that we're using, which in this case was I2959, this being our first piece of work. And what's important is actually the overlap between these two curves. So that can tell you how much light energy is actually being absorbed by that solution. And that's important for characterizing some other properties, which I'll talk about in a minute. Then we set up another experiment where, to really understand what's going on when the light hits the photoinitiator, what we can do is we can repeatedly expose the photoinitiator solution to a light, the same light source we've characterized, and we can monitor the change in, or the degradation of that photoinitiator as it degenerates from the red curve, which is the original initiator, and the blue, which is the degradation byproduct to after it's been cleaved. So using this kind of approach, we can actually calculate properties such as the molar extinction coefficient, and knowing the absorbed energy which we get from the overlap in the first graph, we can also calculate the quantum efficiency, or which is the proportion of molecules which when hit by a photon actually generate a radical. So this might all sound kind of quite academic, but using these numbers, you can actually work out things like what is the free radical flux. That means how many radicals are being generated per volume per second at a particular light intensity or concentration. This analysis is all done without having any hydrogels involved just yet, just to understand the system. And I'm opening with this work because I see that this kind of analysis is, I think, rarely performed in the literature, and I think that's a shame, because it's useful and it can help you gain some insights into your materials. So let me give you an example. A couple of years after we published that work, I saw a great talk by Natalie Bock from QUT, and she presented some data like this, which showed, which was generated by her student Margot. They were adding cefazolin, which is an antibacterial drug, to GELMA, and seeing a reduction in stiffness, but they didn't have a satisfying explanation as to why. So we started working together, and I did some in situ photorheology experiments to try and understand what was going on. You know, this drug molecule, how could it be influencing the cross-link density or the stiffness of the hydrogel? So we did some in situ photorheology and found that the rate of the reaction was totally different across samples with different amounts of the drug. And as you add more cefazolin, the reaction rate drops. It was quite a dramatic drop. And you know, why? Well, the question is why. So one idea might be that the drug is absorbing light and then therefore reducing the light intensity. But this couldn't be the case in this case, because the drug doesn't absorb at that wavelength. You can see here the UV spectrum of the cefazolin, and the 365 is around here, so there's no absorption. But another idea is that the cefazolin might be actually quenching the radicals that are being generated when the photoinitiator is split. So looking at the rate of reaction equations, which I had on a couple of slides ago, you can kind of test that hypothesis, because if that's the case, if it's the case that the drug is quenching, then you'll know that the reaction rate should drop in proportion to the square root of the drug concentration. So to test that hypothesis, that's what we plotted, and we saw a nice agreement. So plotting the reaction relative cross-linking rate against the square root of the drug dose, we see that it does agree with that square root dependence. So that supports the idea that the drug is actually quenching some of the radicals and then therefore slowing down the reaction rate, and that's why that drug-loaded material was softer than the non-drug-loaded material. But we can go a bit further. So using the characterization that we've done before in terms of the number of radicals that are being produced, you can actually work out if this is what's happening, how much of the drug is being consumed. And you do all the calculations, you know, in molar values, and you can actually, you know, the risk is that if your drug is quenching the reaction, then maybe all the drug is getting used up during the reaction, in which case you won't have any left over to release to deal with the bacteria. But it turns out that when we run these calculations, we see that only you know, a couple of % of the drug is actually being used up in this quenching process, and the proportion actually changes depending on the concentration. So this is just an example to show that if you apply like this quantitative approach based on kinetics, you can gain some insight into what's going on within your material as it's cross-linking, and help, you know, solve problems or explain results that you wouldn't otherwise be able to explain. And there's a lot of labs now that are loading growth factors into photocross-linkable hydrogels, or that are studying whether encapsulated cells affect cross-linking. So I think we should consider doing more in situ measurements to answer these kind of questions. So in one more example on this, we had a study where we were also reinforcing GELMA using polycaprolactone, a bit like what Ferry was talking about before. And we were finding that the GELMA was softer than we would expect in the vicinity of the polycaprolactone material. And we knew from the direction of the light that it wasn't a shadowing effect. It wasn't like the light was blocking or the PCL was blocking the light, and that was causing the softness. So what we did was we set up this experiment, an in situ photorheology experiment, where we coated the rheometer plate in PCL, and then and the light is being shone in from underneath here. In this case, we're using LAP and 405 nanometer light. And what we did was we turn on the light, and when it's on continuously, like with and without the PCL plate, we see that the stiffness evolves very similarly in each case. So basically, it makes no difference if the PCL is there when the light is on continuously. But the behavior is very different if you turn off the light after just 60 seconds. So in that case, there's this is a free radical polymerization reaction. The radicals are live, they're moving through the gel. The polymerization continues for quite a long time, even after the light is turned off. But the reaction stops a lot faster when you have a PCL plate rather than when you have just a normal steel plate. And so this kind of supports the idea that the PCL is actually quenching some of those radicals and then slowing down the reaction. And in our normal case, we actually only use a 60-second light exposure. So if that's the case, this supports the idea that the PCL is actually quenching some of those radicals and stopping that reaction faster than it would otherwise. And that's why we were seeing softer material only in the vicinity of the PCL material. Yeah, so another example of how this technique can give you new insight. That question of cytotoxicity always kind of comes up, and it's a question that we've looked at ourselves in photoinitiated or photocross-linkable hydrogels. In our case, we've looked, we've done experiments studying what happens to our cells. We use mesenchymal stem cells, and what happens when we expose to UV alone, to the photoinitiator alone, or to the photoinitiator plus the light. So in this case, it's actually a 400 nanometer light, which is not technically UV, it's a bit on the cusp. So anyway, we found that by far the most toxic element was the free radicals produced when you have the combination of the photoinitiator and the light. And in our particular experiments with our particular cell type, we didn't see any noticeable loss in cell viability as measured by metabolic activity when we shone the UV alone compared to control. But coming back to this free radical generation, it's not just the presence of free radicals. Obviously, the dose makes the poison. So we've studied before also, we can calculate, like I said, the free radical flux, and we know that when we do that kind of experiment where we control the rate of free radical production and assess how that's affecting cells, we find an obvious and huge difference where the lower light intensity results in much lower loss of cells, even when like the eventual stiffness of the materials is controlled. So another reason to use very low light intensity, the lower the better. You reduce that free radical flux, and that is the most toxic element that's affecting the cells, at least in our experiments. Okay, so I'll move quickly now because I think we're getting a bit late. We've also looked at sterilization methods. We've assessed various kinds. Everything on this slide we've tested apart from supercritical CO2, which I think is an interesting one. But the big problem is, how do you sterilize large amounts of material? Typically, when people synthesize small amounts in the lab, they might do a filter sterilization, but we want to scale up to multi-kilo batches, and that's not quite possible. And it's also not possible to do that when you've got large molecular weight materials like hyaluronic acid which might be 1 million Dalton, right? So in our experiment, or in this paper that I'll describe, we assessed 4 of these sterilization methods. We assessed them for their effect on viscosity, on molecular weight changes, and so on, degree of functionalization changes due to the sterilization effect. We checked whether printability was maintained, and also cell function in terms of differentiation towards production of neocartilage. And we also validated the sterilization methods by deliberately inoculating with bacteria, sterilizing, and then testing whether there was any bacteria left over. And the take-home message is that in this of those that we tested, ethylene oxide gas, which is a commonly used in the medical device industry, was very promising for bulk sterilization of these hydrogels, and the gels that we tested, or the lyophilized powders that we tested, were Gelma, HAMA, HA, and gelatin itself. So we tested all of those under all of these conditions. Okay, finally, I'll just quickly introduce another project we have, which is to develop a new fabrication method based on fused deposition modeling. So typically, these printers, these FDM printers, are used in high schools. They're cheap, they're easy to use, and so on. But they're not so much used in tissue engineering and biofabrication, because resolution is poor, and you can't print very complex structures. So what we do is we print with a water soluble material, and we design we print a mold. Basically, we print a mold that has cavity inside, defining the shape that we want to get in the end. This is what it looks like kind of mid-print, and all these holes there correspond to empty spaces, which is going to be filled up with a casted material later on. And within any empty space in a mold, you can actually define pretty complex structures. So this is an X-ray, CT scan that's showing the empty space within such a mold. And basically, we develop an injection molding process where we screw in a syringe containing our material, and then inject the material. So first step is to print the mold. Secondly, we cast in what we want, and then we dissolve the mold away. This is polycaprolactone again, creating a human scale ear structure. Because the spaces between the lines can be defined, it can be any kind of distance. You can get higher resolution than just than defined by the nozzle. You can create complex structures, again using polycaprolactone or acrylic or silicone, even hydrogels as well we've used for this process. And one final innovation is you can use a process called gas-assisted injection molding, where you kind of fill up your mold, then blast out the insides, and then you get hollow tubes, hollow branched tubes, which we've developed into a hydrogel branched vasculature for seeding with endothelial cells. We can control the thickness of the walls. And what we actually do for the seeding is we have multiple layers. So we have an optimized layer on the outside for stiffness, and an optimized layer on the inside for cell attachment, a bit like Ferry was talking about. And this is ongoing work. So thanks very much for your attention. Most of the work was done by Steph and Kate, and the early lab work by myself and some other students. Thanks to everybody, and please visit us if you're in Melbourne anytime.
Thanks so much, Cathal. Just waiting for my camera to start again. Fantastic presentation, really nice work. I really appreciate you being here today and sharing your work with everyone today. So again, just to all attendees, if you have any questions, please feel free to type them out. There's a few already. One of them is from your co-worker Natalie, who's saying, thanks for a great talk, Cathal. Can you explain how do you explain that visible light cross-linked with LAP causes more toxicity than UV at 365 nanometer? Is this consistently seen across different types of cells?
Maybe I gave the wrong impression with the slides or what I said. We haven't specifically compared 365 to 400. I would not expect, I mean, based on the UV energy, you obviously expect the higher wavelengths to be more safe. Just the main point is that the light itself, with no photoinitiator, in our experiments was relatively benign, and it was the combination of the light with the photoinitiator, creating those radicals. It's the radicals that we identified as being the most toxic, and that could be the case for any wavelength of light, really. Which anytime you're generating radicals, which is for any of these hydrogels, the radicals we suppose will be the most damaging.
Yeah. A few maybe follow-up questions regarding that as well. Do you anticipate, for example, that pure UV exposure, so without the presence of a photoinitiator, could actually lead to like long-term effects of cells? For example, they might have, let's say, the similar metabolic rate or proliferation rate, but maybe it could change things that you can't directly measure using these methodologies, DNA changes, things like initiation of cancers and things like that. Can you maybe comment on that?
Yeah, that's definitely the case. And this is a wavelength dependent effect. So we explored that topic in detail in our paper on the cytotoxicity and genotoxicity of photocross-linking reactions, led by Serena Dukey, and it was in Stem Cells Translational Medicine a couple of years ago. And we do look at the effects on DNA, and as well as methods of characterizing those effects, because it's true that in the field we often focus too much on just viability or metabolic activity, and obviously that's not the whole story.
Yeah, so there's a lot of interest in that topic here. So another attendee says, thanks for a nice talk. Did you look at DNA damage markers like CPD in the presence of UV or 1.5 nanometer light in the cells?
Yeah, so that work is ongoing right now. It's not ready to talk about, but yeah, we are conducting those experiments.
Fantastic. I also had a follow-up question, if you don't mind. The data you showed basically with the metabolic activity that also Natalie referred to, were these measurements done inside of a hydrogel? So basically in the presence of functional groups that are cross-linked by the radicals, or did you have basically cells swimming in a solution of photoinitiator and then you expose them?
Yeah, we've done both. I mean, we've done the 2D controls as well as within the hydrogel. It's harder to, yeah, I mean, it's harder to decouple in that case, the different effects. The results on the particular slide that I showed were just the 2D ones, but the take-home message is similar. That in our experiments, we found that the radicals were the most damaging. If you work out, when you generate the free radicals, if you work out, what percentage of the radicals are actually being used to generate, to react with GelMA, it's usually only about 2 or 3% that are actually used to start the reaction. The rest of the 97% of the radicals are potentially those toxic elements.
I believe also Professor Travis Klein's group has done a little bit of work in that space as well. And I think what they were able to at least get some preliminary evidence for is that the presence of these, let's say, unreacted methacrylyl groups actually acts as a cytoprotectant. So the explanation, I believe, if I remember correctly, was they have relatively low activation energy. So as energy comes in, cleaves the photoinitiator, that's a highly reactive system that then preferentially also reacts with these unreacted functional groups. So these can basically, to some extent at least, shield the cells from damage. So I'm wondering, is that something you've observed as well? This is kind of a similar question to my previous question, but maybe you can.
Yeah, no, it's not something we've looked at specifically, but it does sound absolutely plausible. Yeah, that sounds like great work.
Cool. Last question for today. So the ETO gas, you did mention that's a great sterilization method, and I do remember also seeing your paper, so really nice work there. Can you comment on any potential changes in, for example, molecular weight, things like that? I know you showed viscosity. Have you looked directly at molecular weight changes? Have you seen a degradation?
We did look at molecular weight changes. We saw a small decrease with ETO, and like it went from about 74,000 down to 70,000 Dalton. So it was a small decrease, which is actually, it was comparable to what you get when you keep the material at a warm temperature over time, just through hydrolysis. But there was a huge change when you autoclave it. Obviously, a huge drop in the molecular weight as comparison.
Okay, no, fantastic. Also, even you showed with filter sterilization that preferentially the larger molecules are being retained in the filter, which obviously makes sense, and the overall average molecular weight also decreases. So certainly something that people and users of GelMA should think about. I think there's also a lot of protocols out there that recommend dissolving GelMA at, let's say, 60 degrees. Of course, it is much quicker than 37 degrees, but it can also hydrolytically and thermally degrade the material and lead to unforeseen reduction in molecular weights there as well.
Yep.
Fantastic. I think there's no more questions from the audience. So thanks so much, Cathal, for your presentation. Really great to have you here today. Ferry, if you don't mind just maybe turning on your camera one more time, that'd be fantastic, just to say thank you to everyone. And Cathal, can I just ask you to close your presentation now, or maybe I can actually turn this off myself. Oh, here we go. Perfect. Thanks so much. So again, Ferry, Cathal, I can't even express how thankful we are to have you here today. Really fantastic presentations. I think everyone really enjoyed it. We had really good engagement from our attendees as well. So thanks again from everyone, our Gelomics team, and also everyone on the audience here today. So also big thank you to our global audience today. I think we've had a lot of attendees all across the world. So thanks so much for joining in into our webinar today. So please stay engaged with us on LinkedIn, and don't forget to also sign up for our monthly newsletter on our website. And also a recording of today's webinar will be sent out to all the participants and will also be made available on our YouTube channel. So please, if you want to, have another look and look into some of the details, feel free to check that out on the YouTube channel as well. And of course, there will be new webinars coming up in 2024. It's been a fairly successful, I believe, series this year with lots of attendees and audiences across the world. So we'd like to keep that going next year. So before we conclude, really, if any of our attendees have feedback and like to share some of the thoughts, please feel free to send us an email on to info@gelomics.com. Let us know what you think, what we could improve, and which topics you'd like to have covered in an upcoming webinar, or potentially even which speakers we should think of inviting for next year. So yeah, with that, thanks again to everyone. We're really looking forward to meet or see all the attendees in the next webinars as well. And we really hope you enjoyed the session just as much as we did. So yeah, thanks again. Wishing you all a wonderful holiday season, a nice, good start to the next year ahead, and see you all in the next year. Thank you, everyone.
Thanks, Christoph.
Thank you.
Thank you. That was great.